We are going to start the webinar and myself, Fathima Beegum, research scholar working in Manipal College of Pharmaceutical Science. Thank you, Meg, for the kind words. We are going to start the webinar. The webinar is about the technology. Which technology? It is about CRISPR technology. It is a groundbreaking or breakthrough in biomedical research. So today we are going to discuss about what is this technology, what is the future of this technology. We know that millions of people, they are suffering from various sufferings such as diseases such as cancer, then children with AIDS, cystic fibrosis, sickle cell anemia, beta thalassemia. Just imagine a condition for all this disease, a cure, a solution, just by rewriting the genetic code present in their genome. That is the promising of CRISPR technology. So coming to CRISPR, CRISPR is actually a gene editing technology. What is CRISPR? So when you need to know about CRISPR technology, first you have to get an idea about what is gene editing. What is gene editing? Gene editing means it is a method in which the genes present in the genome sequence of a living organism is edited. Edited means it is deleted, inserted, or base pair substitution is done. So that is known as gene editing. And very interesting actually this gene editing. And one more point, from the time onwards, Watson and Crick, they discovered the double helix structure of DNA. There are so many scientists conducted so many researches regarding to know about the importance of DNA in the genetic of the organism. And CRISPR is the latest technology in it. So gene editing, if you're saying about gene editing, the generation of gene or development of gene editing, it is classified into 3 generations. The first generation, second generation, and the third generation. The first generation is zinc finger nucleases. It is endonuclease enzyme, which is used for gene editing, for cutting the genes present in the genome. Then the second generation is the transcription activator like effector nuclease, which is known as TALENs. And the latest generation or the third generation is what we are going to discuss today, and that is CRISPR and CRISPR associated protein 9. Precisely, we are saying it is CRISPR-Cas9. And what is CRISPR? It stands for clustered regularly interspaced short palindrome repeats. So, knowing about CRISPR or saying something about CRISPR, that is, actually, when a bacteria, it is infected by a virus. Just consider it is bacteriophage. So once it is infected by a virus, this bacteria has a very good mechanism in incorporating the particular segments that is present in the virus in their DNA in a particular pattern, and this is known as CRISPR array actually. And this CRISPR array, it acts as a remembrance for this bacteria. The next time when the same virus attack the bacteria, it will remember the virus. And what it will do, it will release certain enzymes known as Cas9. And this Cas9, it will recognize the particular sequence that is present in the viral DNA, which is known as PAM sequence, protospacer adjacent motif, it is known as. So it is recognized. And what did this bacteria do? Bacteria will cut the DNA of virus with the help of a RNA that is produced from the CRISPR array. So that is the main mechanism how this CRISPR technology is working. For the CRISPR technology to work, what is needed? An enzyme along with enzyme Cas9, which is an endonuclease that is present in the bacteria. Along with that, a guided RNA is also needed. So that too consists of CRISPR Cas9. So it is a natural defense mechanism which allows the bacteria to detect and destroy the virus and attack them. And as we already said, it uses Cas nucleases enzyme that can bind and create double-stranded breaks in the DNA. Again, this Cas9 activity, this enzyme activity, it is an endonuclease which is present in the bacteria, and its activity is depending upon, as I said you before, it depends upon a guide RNA, which guides which segment has to be cutted. That is guided by this guide RNA, as the name suggests. And there are, in case of Cas9, there are two RNAs present, CRISPR RNA, what is known as CRRNA, and also a trans-acting CRISPR RNA, which is known as tracid RNA. So this CRRNA, it is actually complement to the viral spacer, which is stored in the original infection. The what is spacer? It is the portion of virus that is stored in that gets incorporated in the bacterial cell. Okay, that is known as spacer. And what happens in complementary to the spacer, a RNA is there, which is known as CRRNA, and tracer RNA, it acts as a scaffold, and both of this, it consists of the guide RNA. So CRISPR to work, it needs an enzyme. That enzyme break the DNA strand which is present in the virus with the help of a guide RNA. So scientists have conducted so many research from 1983 onwards. They are conducting research to know about whether this property can be taken, whether this property of bacteria to recognize the RNA and cut a particular sequence can be taken and can be used for gene editing purposes. And there comes the CRISPR technology's initial stages. This is what is occurring in bacteria. You can understand by this figure, I think you can understand by this figure. Just consider this is the bacteriophage which infects the bacteria. This is the bacterial cell inside. So what happens is a virus with unknown genome, it attacks the bacterial cell first. A portion of this viral DNA sequence, it is copied to the new spacer that is present in the bacteria. You can see this blue color spacer. Spacer is incorporated in the bacterial DNA. Then what happens? The bacteria now contains the spacer. So next time, what happens? This virus is with a non-genome, already non-genome. This non-genome, it attacks the same bacteria. What happens is that the genome is recognized by the CRISPR RNA. CRISPR RNA will recognize the genome, and with the help of Cas9 enzyme, it will break the viral particles, break the viral DNA, and thereby deactivated the viral DNA. This is what is occur in the bacteria. Just whenever a bacteria is infected with a virus, a portion of it gets incorporated into the viral into the segment of DNA segment of bacteria, and that is known as in a particular pattern, and it is known as spacer, and that pattern is CRISPR array. Then what happens is the next time when the bacteria again infect the virus, what happens is that the CRISPR RNA, it detects the target sequence and it guides the Cas9. What is Cas9? It is an endonuclease. Cas9 to break the viral DNA in the target sequence, and the virus get inactivated. So this principle is taken for CRISPR Cas9 technology for creating double-strand breaks in the DNA, and it took advantage of cellular DNA repair. What happens after this double-strand breakage occurs? That is, I'm saying about the technology. So CRISPR, we have just selected one host organism, and we are going to place the CRISPR, that is Cas9 enzyme, along with the guided RNA, that is the CRISPR system in that organism. So what happens is when the guide RNA, it detects the correct sequence of the DNA, it will the double strand will be broken, and what happens is after that the process is known as cellular repair occurs. That is the natural cellular repair occurring in the organism itself. It is of two types, the cellular repair, and that is non-homologous end joining and homology directed repair. Through, that is, the organism or any living organism has two DNA repair pathways mainly. So once the DNA strand is breaking, what happens? Inside the cell, the DNA repair mechanism takes place, either non-homologous end joining. What happens in non-homologous end joining is, it is error prone, that is the insertion, deletion, etc. are occurring in non-homologous end joining repair pathway. And also if this pathway it is taking place in the code, that is the genetic code, the code which codes for the DNA or genetic material, then what happens is that frameshift mutation occurs, and what happens, the gene became inactive. So one point just take into consider here, that is the knockout gene knockout can be conducted by using non-homologous end joining repair process. Coming to homology directed repair, it is an actual repair process. We can insert new genes by using this repair process. New genes can be inserted. That is what is explained in the last point. NHEJ, it renders the genes non-functional. So knockout models can be created by using NHEJ mechanism, repair mechanism, while HDR, it is exploited to insert new genes. The desirable gene, the which gene we needed, that desirable genes can be inserted into the genetic material by using the HDR method, repair method. So this is what is known as CRISPR. In CRISPR, we know that one SG, single guided RNA is present. Okay, single guided RNA is present, and this is the spacer that is present in the CRISPR, and the target DNA is there. When this RNA guides the region, what happens? This particular region, breaking occurs. So DNA double strand gets break. This is the center. This is known as protospace adjacent motif. It is the region where this guided RNA detects the specific segment. And what happens after that? Two repair mechanisms can occur. Either NHEJ occur. So what happens? Insertion or deletion occurs, or HDR occurs. It can lead to precise editing. So once more, how CRISPR works, if we are saying, the Cas9 protein is there. It forms a complex with the guide RNA. So Cas9 along with guide RNA is there. This complex, it attached to the genomic DNA sequence that is present. That is already spacer containing genomic sequence will be there. In that genomic sequence, at the PAM region, this Cas9 along with the guide RNA will attach. And what happens in the third step? The Cas9 RNA complex, it cuts the double-stranded DNA. It cuts the double-stranded DNA, and also this is our programmed DNA, the new DNA which we have, which we need to insert into the organism. So what happens? The programmed DNA, it gets inserted at the cut position, and we can insert this back by using suitable vectors to the organism, living organism. So this is how CRISPR works. Then the pioneers in revolution of CRISPR, if we are saying, CRISPR as it is known today, it was developed by two scientists. You may be knowing them. They received the Nobel Prize for Chemistry in 2020, and that is Jennifer Doudna, PhD. She runs a lab at the University of California, Berkeley, and Immanuel Charpentier, PhD, Scientific and Managing Director of Max Planck Unit for Science of Pathogens in Berlin, Germany. They were awarded Nobel Prize in Chemistry for 2020. Then if we are checking the timeline of CRISPR, the establishment, application, and clinical trial gene therapy, if we are looking into that, we can say that it starts, as I said you before, it already starts in 1987. In 1987, the CRISPR clustered repeats in E. coli was identified. Then in 1993, CRISPR clustered present in Mycobacterium tuberculosis was identified. And in 2000, CRISPR families been widespread in bacteria, it was discovered. And in 2002, the term CRISPR was coined in 2002. Then 2005, Cas9 identified this as an endonuclease. It is identified as an endonuclease. And in 2010, detailed role of this CRISPR in bacterial immune system was found out. Then the CRISPR Cas9 gene editing approach publications was there in 2012, and in 13, CRISPR-Cas9 used to edit the specific genes. Then in 2017 was the first CRISPR clinical trial, and it was for HIV, HIV treatment it was for. And also in 2017, the first CRISPR germline editing in implanted embryos also took place. Then in 2019, first in vivo CRISPR clinical trial. It is for the treatment of blindness. And 2018, for cancer therapy, CRISPR has been introduced. Then, as you know, 2020 is the time where our scientists Donna and Charpenter, Dr. Donna and Dr. Charpenter, they received the Nobel Prize. 2020 itself, in vivo CRISPR clinical trial for blindness has been introduced. And in 2021, so many clinical trials for different disorders such as sickle cell anemia, beta thalassemia, then muscular dystrophy, etc. So many clinical trials have been introduced. In 2022 US trial, the first participants for the clinical trial with HIV, infected participants, was done in 2022. And a very important achievement of CRISPR technology is in November 2023, and that is gene editing tool. It scored the first clinical approval by FDA for treating sickle cell anemia and beta thalassemia in United Kingdom. After weeks later, the Food and Drug Administration in US, they greenlighted this therapy for sickle cell, and is set to rule on beta thalassemia by March of the next year. That is this coming year, March 2024. So that was a groundbreaking discovery, or the main, an important step in the CRISPR technology. FDA approved the therapy for sickle cell anemia. Then coming to discuss about the protocol of the CRISPR, you may be knowing the first point is to choose which Cas enzyme. As I said you before, CRISPR is always associated with a Cas enzyme, and precisely we are learning about, or more research has been conducted in CRISPR Cas9 family. That is a type 2 Cas family member protein, I mean endonuclease. Besides this Cas9, another Cas enzymes are also there. Cas12A, 12B, then Cas-S, etc. are there. But the thing is that for mammalians, that is for humans, more research was conducted in Cas9. That's why we selected, we used to do more research by using CRISPR Cas9 technology or Cas9 enzyme. So the first step is to choose which enzyme is needed. And for choosing which enzyme is needed for our study, we need to take into account the site accessibility. That is, we know PAM. I already said protospacer adjacent motif. It is the region which is present in the host or the host person or the host patient, in which the particular guided DNA has to guide, and the particular enzyme has to cut the DNA. So that region site accessibility is the first criteria for choosing the Cas enzyme. Then second is off targets. What is off targets? That is the other regions which we are not targeting. That regions also have to be taken into account while you select a particular Cas enzyme. Then also on targets, which target we have to cut or which target we have to edit. That also we need to consider. Then the second step is design the guide RNA. The guide RNA has to be customized. So many research tools are available, designer tools are available. By using that, the complementary to the DNA sequence which we needed, we can customize the guide RNA. So guide RNA is designed. After the designing of guide RNA, now guide RNA is there, the Cas enzyme is there. So CRISPR is there. Then this has to be delivered into the host organism. How this has to be delivered into the host organism? Can anyone say? You can unmute and say how this can be delivered to the host organism. This Cas enzyme along with our guide DNA is there with us, but this has to be delivered to the host organism.
By using a vector.
Yeah, that's correct. By using a vector, and that vector can be either a viral vector or a non-viral vector. Viral vectors, the common viral vectors we are using for this CRISPR technology is adenovirus, adeno-associated virus, lentivirus we are using. But as you know, if you are using virus, there are so many advantages as well as so many limitations are there. Advantage is that it will easily pass the cell membrane and reach inside the host organism. Then it does not causes any change in the target sequence, or it does not degrade the target, the transgene. It does not cause any change. But it is also associated, these viral vectors are also associated with certain drawbacks. And the main important drawback is the immunogenicity. That is, it once reaches into our system, it has the ability to rise the immune response and can cause various reactions. So that is a very important problem, and that is the only problem uh, with viral vectors. So since this problem is there, they have been selected so many other techniques, that is non-viral techniques also has been introduced, and such as a complex is formed with liposuction, then electroporation techniques, then complex with different polymers has been done, I mean has been produced, and it has been insert into the host organism. And after this delivery, the next step is repair. Repair process, as we said, two repair processes taking place. Either it is homology, no? Either it is homology joint method, repair system, or non-homology joint method system, or homology dependent repair. Two types of repair can occur. We can insert or delete the DNA or the segment which is needed, and we can also make it knockout. Knockouts can be also produced. Then the last step is the analysis. For analyzing, we know that the technique which we used are gel electrophoresis techniques. No? So gel techniques can be used. For non-specific analysis, we can use gel-specific analysis technique. But for specific purpose, if you need to know whether it is non-homology repair process or homology repair process, or what happened, whether it is insertion, whether it is deletion, whether it is a base substitution, if you need to know that, then we can go for new generation screening methods, next generation screening methods. So this is what is viral vector which we are using. The adenovirus vector we are using, adeno-associated virus is using, lentivirus is using. And for non-viral vectors, we are using physical methods. First one is micro-injection. That is, a needle is there directly inside the nucleus. The DNA, foreign DNA is injecting. That is micro-injection process. The second one is electroporation. By using an electroporator, the particular DNA, it is inserted into the host cell. Then another method is there, you may be familiar, hydrodynamic injection. It's a process by which it is done in, there are so many research, it is done in mice only. That is, in the tail vein of the mice, injection is given. The nucleic acid is injecting in the tail vein. So as that it can directly enters into the nucleus region of the liver and all. So hydrodynamic injection is there. Then coming to chemical methods, I already told liposomes are there. We know that liposomes is a very good platform for delivery of even drugs. Very good platform there. So it is used for the delivery of the particular DNA segments. Then polyethylenamine, modified polyethylenamine by using plasmids. Then nanocarriers, either nanogels or polymerosomes. By using all these vectors, we can insert the desired DNA or desired sequence or the desired portion into the host organism.